Hello, netizens. Very happy to be with you again. I hope you are doing well. So today we are going to discuss about the Mendel's monohybrid cross. So what is our discussion today? We are going to talk about Mendel's monohybrid cross. So in today's discussion, we will talk about Mendel's monohybrid cross. Mendel's monohybrid cross. So let us see first thing first. Okay, what do we mean by monohybrid? What is the meaning of this monohybrid? Monohybrid. So as we know, okay, hybrid means mixing. So it is a hybrid in one character. So what it is hybrid for? Monohybrid means it is hybrid. It is hybrid for one character. Hybrid for one character. It is hybrid for one character. Like what is the meaning of that one character? So it is hybrid for one character. What's the meaning of that one character? Okay, if one character we will take, let's say character of height. If we will take character of height. So what it is hybrid for? Okay, in that it is hybrid of tall and dwarf. Tall and dwarf. Tall and dwarf. If we will take this character, then similarly if we will take second character, okay, seed shape. If we will take the second character as what? Seed shape. So what it is hybrid for? It is hybrid for round color seed, and it is hybrid for which one? It is hybrid for the wrinkled one. So that is the meaning of this. That means here in this is capital T, small t. In this, it is capital R, small r. So cross which deals. So what is this hybrid? Which will be hybrid for one character. Similarly, what is a monohybrid cross? Cross which deals with the, what is this cross? Cross which deals with the inheritance. Cross which deals with which one? Inheritance. Cross which deals with the inheritance of one character. Inheritance of one character. Cross which deals with the inheritance of one character. That is called monohybrid cross. So now let us see, okay, what and how can we derive different formulae based on this. So in Mendel's monohybrid cross, let's say, okay, first we are taking in monohybrid cross, we are going to take, let's say, this one. Okay, this one is parental generation. Parental generation. So in parental generation, let's say this is male plant. Male plant means what? We are going to take here anther. That means pollen grain, anther. So in this, let's say this plant is homozygous tall. Homozygous tall, because both the alleles, or Mendel never used the word allele, these are factors. So what it is? This plant, it is what? This one is tall, homozygous tall. This one, that means pollen grains which will contain male gametes, these are of capital T type. And this one is crossed with female, that means embryo sac. In ovary, embryo sac. Egg is of what type? It is of small t. That means which one is this? This one is dwarf. So what it is? This one is dwarf. So as we know, okay, these parents, they are representing which condition? They are representing diploid condition. So of course he worked on the pea plant, that is angiosperm, and pea plants are diploid. Right, angiosperms are diploid. So what will happen now? In this process, there will be gametogenesis. What will occur? There is formation of what? Gametogenesis. That is what will occur here, gametogenesis. And as we all know, okay, in gametogenesis there is involvement of meiosis, isn't it? So what is involved there? Meiosis is involved. So what will be? Meiosis is reductional division. What type of division it is? Reductional. So if two factors are there, only one will go in gamete. If two alleles are there, only one will go in gamete. If 46 chromosomes are there, only 23 will go in gamete. So gametogenesis, what will happen here? There will be formation of capital T. So what it is? That means this one is male gamete. What it is? This is male gamete. And towards this side, Similarly, there is formation of small p. So out of two homozygous, out of two, only one will go in the gametes because gametes are haploid, and out of two capital T, only one factor will go into this. So what these things are? These things are gametes. As you know, okay, these pollen grain, they will go through the insects, they will go to the stigma, and there they will reach to embryo sac, and then what type of egg cell is there? Small t type of egg cell is there. Through pollen tube, fertilization will occur, and when fertilization will occur, then there will be formation of seed, and that seed will develop into a plant. So what that plant is called as? That is called as F1 generation. So here these two gametes, they will fertilize, and what will be produced there? Capital T, small t. This one is produced here, capital T, small t. So what type of plant is this? This one is tall plant. And which generation is this? This generation is F1 generation. Which generation is this? F1 generation is this. F1 generation. So we must realize, not simply by looking at this, we should not arrive at conclusion. We should try to understand okay, what is happening here in reality. What Mendel is doing. So you imagine okay, he used to collect pollen grain from the tall plant, and after dusting those pollen grain over the stigma of dwarf plant, then fertilization will occur. Then ovary will change into fruit. Ovary will change into seeds. Those seeds he would collect, and after sowing those seeds, he would observe that all the plants of F1 generation, they are what type? Tall plants they are. So can you imagine the kind of patience which was there with him? Okay, from 1856 till 1863, for 7 years he was doing that work. So see how monotonous it is. But at times we get bored. Very often we say or not, okay, I'm getting bored. But to achieve something of this magnitude, we have to be very focused on that work, and that level of patience is required in order to achieve something. So that requires dedication, and we are not working like forget for 7 years. We are preparing hardly for 1 or 2 years, and in that also, for today only we are learning this. Tomorrow next topic we will learn. Day after tomorrow next topic we will learn. Then how can we say that we are getting bored? Isn't it? So it should be way more interesting than. So Mendel worked for 7 years doing the same work. That's why he is called as father of genetics. So that much level of dedication, that much level of focus we must put in our preparation if we want to achieve desired results, isn't it? So this is F1 generation. From the Mendel's perspective, if we think, so what is the thing? That these plants are appearing tall. He had no idea whatsoever whether they are homozygous tall, heterozygous tall. This plant which is of F1 generation, whether he is tall like this. Now we can see whether he is tall like this or he is tall in this manner, isn't it? So he was surprised. In sexual reproduction, there is a contribution of male parent also, and there is a contribution of female parent also. But in this, female characters are not visible at all. So does that mean these characters disappeared? And if these characters disappear, then all the way it becomes which one? Uniparental, and which is the characteristic of asexual reproduction. So definitely where this character disappeared, whether it is present in this, it is not present here, or where it has gone. So to verify this, what's supposed to be done here? The best way to verify is that take a two plant of the same generation, because if this character is present, then it must be present in the same generation plants. So what he took two plants of F1 generation and crossed them, not the different generation. Two plants of, like, after collecting those seeds, after sowing those seeds, let's say he got 1,000 plants. So out of 1,000 plants, he chose two plants. Those two plants, out of those two plants, one will work as a female plant. That means pistil. Another one, anther. So these both belong to F1 generation, and he crossed them. So because these plants, they are of F1 generation, so that is why he called it what? Self-cross. So this one, now what will be the thing? Self-cross. He did self-cross. So of F1 generation, between F1 generation plants. So if we see this here, in F1 generation plants, if we look carefully, so here, what he did? self cross, self cross, self cross between F1 plants, self cross between F1 plants. So these F1 plants, what type of F1 plants are there? We know that F1 plants, they are which one? Capital T, small t. These are the F1 plants. So here, this is capital T, small t. Let's say it is working like anther, pollen grain, male gamete it would contain. And this one, it is crossed. It is crossed with another one, capital T, small t. Because both are F1 generation plant and it is working like pistil. So it will have that embryo sac, in embryo sac, these type of gametes. So up now, this one is heterozygous condition. So what these are working now? These are working like parents. These are working like parents. And what will happen in them? As we were learning, these are diploid. So here again, there will be which process? Gametogenesis will occur. What will occur here? Gametogenesis. And as we discussed previously, in gametogenesis, there is involvement of meiosis. So here, like in case of us, X and Y chromosome is there in human male. So 50% of the sperm will contain X chromosome and 50% will contain Y chromosome because heterozygous condition is there. So similarly here, these some of the male gamete, they will be of capital T type and another 50% they will be of what? Small t type. Small t type. So what are these? These are male gametes. Isn't it also? These are male gametes. And similarly here, these are male gametes. So what will happen here? In this case, if this X cell will be capital T type and another X cells, they will be of small t type. So what are these things here? These are X cells in embryo sac. So these two type of gamete, one is capital T, another one is small t. Two type in male, two type in female we will get, right? So as you know, okay, this one, what can happen for fertilization? Okay, this gamete can fuse with this one, this gamete can fuse with this one, or this gamete can fuse with this one. So what we will get here? Capital T, that is what we will get here. Here what we will get? Capital T, small t we will get. Isn't it also? Or else what can happen? This gamete can fuse with this one. So what we will get? Capital T, small t. Or this gamete, it can fuse with this one. So what we will get here? Small t we will get. So these are the possibilities. 1, 2, 3, 4. These 4 zygotes we might get. So how many plants we will get? 4 plants. Right? These same things graphically represented by a British scientist called Punnett. So that's why we call it Punnett square. So how do we represent that? So same here. Okay, these things we can represent in Punnett square also. So how it is done? Let us see this. So we have got these one. Okay, this one, 1, 2, 3. These 3 tall and 1 dwarf. So same thing we can represent graphically using Punnett square. So what Punnett did? Punnett, he did which one? See it. So 2 type of male gamete and 2 types of female gametes are produced. 2 type of male gamete and 2 types of female gamete. So in this, in Punnett square, what do? We have this one. So in this, towards this side we write male gamete. Towards this side we write female gamete. So here, what is another one male gamete? Capital T. Another male gamete? Small t. Similarly, here what is the one? XI, capital T. Another one? Small t. So this male gamete can fuse with this female gamete. So what we will get here? There will be formation of capital T, capital T. This male gamete can fuse with this female gamete. So capital T, small t. This female gamete can fuse with this one. So capital T, small t. And this can fuse with this one. So small t, small t. This is which generation we are talking about now? We are talking about F2 generation. Which generation is this? F2 generation is this. So very much similarly, by drawing, it is not taking much time for us. Within fraction of seconds, we are able to do this. But again, you imagine the hard work of Mendel. he used to collect pollen grain, dust over the stigma, and then would wait for months together so that that plant will become mature, seed will develop. After collecting those seeds and after sowing those seeds will form plants, and then he observed. So what he observed, he was very happy. 25% plants were dwarf. Right or no? Which plant is this? This one is dwarf. And this is dwarf. And these three plants, 1, 2, 3. These three plants, 1, 2, 3. These three plants, 1, 2, 3. What type plants these are? These are tall plants. So he was so happy because he did the self-cross between F1 generation only. New plant he did not take. So definitely this dwarf plant, it reappeared. The parental character reappeared. The recessive character reappeared. Recessive character reappeared. Recessive character reappeared here. Recessive character reappeared here. And he was so happy. It did not lost. It was just hidden in F1 plants. So it got reappeared. So now this thing we are observing, three plants are tall and one is dwarf. That means what we are saying is 75% plants are tall and 25% plants are dwarf. So what is the ratio? 3:1 ratio. In which ratio he got these one? 3:1 ratio. So what it is called as? This one is called as phenotypic ratio. So what type of ratio it is? This is phenotypic ratio. Phenotypic ratio of Mendel's monohybrid cross. So now listen here. Genotypic, you see, one type of genotype is this one, capital T, capital T. Genotypic ratio when we talk. Genotypic ratio. So genotypic ratio, what is there in genotypic ratio? One is which one? Capital T, capital T. Then two are what type of plants? Capital T, small t, capital T, small t. Capital T, small t. And another one is what type of plant? Small t, small t. So what is that last one? So what is the genotypic ratio here? Genotypic ratio is 1:2:1, isn't it? 1:2:1. Now these formula you need not by heart. If you notice here, you see, okay, when we are talking about monohybrid cross, half hybrid cross, so we cannot do. Of course, obviously, that is practically impossible. So at least one character you will take in order to conduct your cross. So when we take one character, how many zygote or offsprings we are getting here in F2 generation? So see, zygote or offsprings. If you look at here, zygote or offsprings. Zygote or offsprings. We are getting here what? These things we are talking for F2 generation, F2. So when you take monohybrid cross, 1, 2, 3, 4, 4 will be formed. So what will be the number of zygote or offsprings? It will always be 4 to the power n. Where what is n? N is cross. Whether it is monohybrid cross, 1. Dihybrid cross, 2. So 16. Trihybrid cross, so 3. So what should be the formula in order to find out the zygote or offsprings? 4 to the power n. Where n is which one? What type of cross we are doing? Whether we are talking about monohybrid cross, whether we are talking about dihybrid cross, whether we are talking about trihybrid cross. So that will be the type of zygote and offspring, isn't it? Similarly, if we look at the genotype in F2 generation, if we look at the genotype in F2 generation, so when we are taking one character, that time itself, how many type of genotypes we are getting? We are getting, this is one type of genotype, this is second type of genotype, and this is third type of genotype. These two are the same, these two are same, exactly same. So one type of genotype, second type of genotype, which is of 2 in number, and third type of genotype. That means in F2 generation, types of genotype, So what it should be always? 1, 2, 3. When we take 1, then what is the least number we will get? 3. So again it should be 3 to the power n. 3 to the power n. Where again n is which one? N is cross. Whether we are talking about monohybrid cross, then 3 to the power 1. 3 we will get. If we are talking about dihybrid cross, then we will get 9. 1, 2, 2, 4, 1, 2, 1, 2, 1, 9. Like that we will get. So that is the type of genotype. Again look, when we talk about the phenotype, So how many type of phenotypes we are getting? One is tall and another one is dwarf. Okay, when we take monohybrid cross, then we get two type of phenotypes. Half hybrid, so we cannot do. Definitely that is like practically impossible. So when we take one character inheritance, that time also we will get two types of phenotype. So what is the least types of phenotypes? Similarly, we write phenotypes, type of phenotype in types of phenotypes in which one? F2 generation, types of phenotype. So if we look at here, then what is this? Types of phenotypes. Types of phenotypes. Types of phenotypes. In which one? Types of phenotypes in F2. Types of phenotypes in F2. So what is that? Minimum we are getting tall and dwarf. Minimum. Half hybrid though we definitely cannot do. So that will be 2 to the power n. What is that? 2 to the power n. And where n again is what? Cross. Where again is what? Cross. So if it is monohybrid, cross, then what will be there? 2 to the power 1. So how many types of phenotype? 2. If it is dihybrid cross, then what it should be? 2 to the power 2. So what? How many types of phenotype we will get? 4. 9 is 2, 3 is 2, 3 is 2, 1. So 4 types of phenotypes we will get. If it is trihybrid, then 2 to the power 3. And we must remember all this we are learning based on Mendelism, right? So there is no need to by heart all these formulas. Definitely we can remember, we can derive based on this from monohybrid cross. So these are the ratios and this is how we can find out, right? Similarly, if you see in monohybrid we took one heterozygous, capital T, small t. So what will be the types of gametes? Types of gametes will also be 2 to the power n, right or no? Where n is what? Number of heterozygous. So what is the another thing? Another thing is types of gametes in F2 generation. So types of gametes, what will be the thing? Types of gametes. Types of gametes will also be 2 to the power n, where n is what? Number of heterozygous. What is this? Number of heterozygous. So based on this, you can solve these questions. Like practically we take for dihybrid cross, if I hope you are clear with this. So what is the types of gametes? 2 to the power n. What are the types of phenotypes? 2 to the power n. Types of genotypes? 3 to the power n. Zygote or offsprings? 4 to the power n. See, we will verify this. So types of gametes? 2 to the power n. Types of phenotypes? 2 to the power n. Genotypes? 3 to the power n. If you have doubt, you can pause and revise. So let's see this. What we are talking about? Few formulae which we have derived. Okay, types of gametes 2 to the power n. Similarly, types of phenotypes. Types of phenotypes 2 to the power n, which is cross. So first thing, second thing, third one. What is third one? Third one is types of genotypes. Types of genotypes, which we saw just now. So types of genotypes will be 3 to the power n, right? Then what was that? Zygote or offsprings. Zygote or offsprings in F2. So what will be zygote or offsprings in F2? 4 to the power n. Let us verify all this for dihybrid cross. For which one? For dihybrid cross. For dihybrid cross. So what is dihybrid cross? 2 heterozygous. So if we will take capital R, capital Y, small y, right? So how many heterozygous are there here? 2 heterozygous. 2 heterozygous. So what will be types of gametes? First one, types of gametes. Types of gametes will be 2 to the power 2. That means how many types? 4. What will be the second one here? Types of phenotype. So types of phenotype again. Types of phenotype. So type of phenotype in this is again 2 to the power 2. Dihybrid cross it is. So what is that? 4. And this 4 you know, 9 is 2, 3 is 2, 3 is 2, 1. Round and yellow. wrinkled and green, round and green, wrinkled and yellow. So you will get 4 types. Similarly, third one, if you look at, okay, third one, what is third one? Types of genotype. Types of genotype. Types of genotype in this. So types of genotype will be 3 to the power 2. That means 9 types we will get. 9 types. What is that? 1, 2, 2, 4. 1, 2, 1, 2, 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 types of genotypes we will get. This I will explain you in our dihybrid cross, forthcoming lecture. So this is third one. And what is fourth one? Is zygote or offsprings. So types of zygote and offsprings in F2. So what we will get here? We will get 4 to the power n. So which cross is this? Dihybrid. So how many we will get? We will get 16 offsprings. Correct or not? 9 is to 3, 9 plus 3 plus 3 plus 1. So how many offsprings we will get here? 16. Similarly, in trihybrid cross, if you will see, if we will verify, then we will get 64. We will get 64. Thank you all. Be wise and revise. For that, you must know revision is the key. So be wise and revise. Thank you all.